in this module we'll study about the cell organelles, which are very important for the cell to perform the various functions, and these cell organelles are embedded inside the cytoplasm. Students, these cell organelles performs different functions. Like the first one we have is, it has to synthesize the substance. For example, protein synthesis is done by ribosomes, lipid synthesis on the surface of SCR, and photosynthesis of food is done by the organelle, that is the chloroplast. Second we have is, it has to secrete cell products, or we can say, for example, we have enzymes, hormones, mucus. So all these things, they are basically secreted by the cell organelle. Next we have is, it has to digest those substances which are taken up by the cell during endocytosis. Such intracellular digestion is done by the enzymes of lysosomes. And the last one we have is, students, it has to generate some energy. Now firstly we'll be starting up with the cell organelle, that is Golgi apparatus, students. You all can see here, this is a complete structure of Golgi apparatus, which was discovered by Camillo Golgi. Now you can see here, students, this is the diagram of Golgi apparatus. Fine. It consists of a set of membrane bounded fluid filled vesicle. You can see these are the cisternes. You can see, students, on the right hand side, these are the cisternes. Fine. And they form a stack like structures which are parallel to one another. You can see here, this is a lumen. Lumen is a cavity, students, which is formed inside these cisternes. Now you can see here, these are the vesicles. Fine, small vesicles you can see here. These are fused together to form these cisternes. Fine, and basically this is the cis phase. Now after forming up these cisternes, finally the vesicle, they get discharged. Fine, and this is what the maturing phase is. Fine, students, and the function of these vesicle, or we can say the secretory vesicle, is to secrete some enzymes, proteins, and whatever is needed up by the organism. Now moving on to its nature and occurrence. It consists of membrane bounded fluid filled vesicle and flattened cisternes. We have already discussed these things, and these cisternae, they are stacked together in parallel rows. Now students, Golgi apparatus exists as an extensive network near the nucleus in the animal cell, but in plant cell it contains many freely distributed subunits called dictyosomes. So if this thing comes in your examination, that what do you mean by dictyosome, so you can write down. This is basically the Golgi apparatus only, students, but in plant cell they contain like freely distributed subunits. So in plant cell this is termed as dictyosomes. So in this point you can read it that Golgi apparatus exists as an extensive network near the nucleus. Fine, so it is present near the nucleus, students. Like you can see here, fine, these are the Golgi apparatus which is formed, or which are basically present near the nucleus. Now cisternae are formed at one end of the stack, and this is called the cis phase of Golgi. ये चीज मैंने आपको already show कर दी है, students. You can see here, this is the cis phase, fine, where these vesicles are formed. They are budded off as vesicle at the other phase of Golgi apparatus, and that is called the trans phase of Golgi. This thing we have already done, students. See, this is the diagram. बहुत सारी चीजें diagram से ही clear हो जाती हैं, students. So you can see here, this is the maturation phase, students. Fine, जहाँ पे जितने भी आपके vesicles हैं, they actually become mature, and finally they are budded off. And these Golgi apparatus, students, they are absent in bacteria, blue-green algae, mature sperms and RBC. अब exactly आपने last में जो line दिख रही है आपको, students, ये बहुत ज़्यादा important है. Rough endoplasmic reticulum give rise to smooth endoplasmic reticulum, which forms the Golgi apparatus. Now this was all about its structure. Moving on to its functions. Now students, the main function of Golgi is secretory in nature. It acts as an assembly area for the storage, processing and packaging of cellular secretion. So students, whatever is being secreted by the Golgi apparatus, it is being packed up and stored and finally processed. And it packages materials synthesized in the cell and dispatches them to target sites such as we have plasma membrane and lysosomes. So anything that is being formed, like we have, will be studying later on about the endoplasmic reticulum, students, which forms or helps in forming the proteins. Finally, these proteins are being stored, processed and packaged up with the help of the Golgi apparatus. It also produces secretory vesicle which contains cellular secretion. Now here students, what exactly do we mean by the secretions? It can be anything. For example, we have enzymes, we have proteins, we have cellulose, etc. It is also involved in the synthesis of cell wall, plasma membrane and lysosome. So students, it's very important thing that you all need to memorize is here that this Golgi apparatus helps in the synthesis of cell wall, fine, in case of plant cell, plasma membrane in case of animal cell, and also helps in the formation of lysosomes, which we'll be studying later on. So this was all about the Golgi apparatus. Now moving on to the next organelle, students, that is the endoplasmic reticulum. Fine. You can see here, this is the complete structure of endoplasmic reticulum. So this is the nucleolus. Fine. This is nucleus. So nucleus basically किस चीज को embedded कर रहा है students यहाँ पे? Nucleolus को. So this red like structure is nucleolus. Then we have this nucleus and you can see here finally this endoplasmic reticulum is connected with this nucleus here. Fine. So these are the rough endoplasmic reticulum, students, and you can see here that on the surface of rough endoplasmic reticulum we have this tiny dot like structures. Fine. So these structures are nothing but they are known as ribosomes, students. And always remember one thing, that these ribosomes are only present on RER. They are absent in SER, that is the smooth endoplasmic reticulum. You can see here, students, these are the smooth endoplasmic reticulum. And these RER synthesize, basically they are having ribosomes. So the function of ribosome is to synthesize the protein. Now students, endoplasmic reticulum, it is basically located around the nucleus of the cells. They are large network-like structure. It looks like long tubules or round or oblong vesicle. So students, this endoplasmic reticulum, it's basically of two types. The first one we have is RER, another one we have is SER. We have already done this thing, students, you can see here, fine. This one is SER with the yellow, RER, and this one is SER without the ribosomes. Now students, here are the important functions of this organelle. The first one we have is that it forms the supporting framework of the cell. It basically gives that structural support. It provides the pathway for the distribution of nuclear material from one cell to another. So finally, it helps in the transportation, students. The next we have is certain enzymes present in smooth endoplasmic reticulum. Now student, this is basically the function of smooth endoplasmic reticulum, that is to synthesize the lipid, steroid, which is an organic substance, and cholesterol. And these helps in the formation of plasma membrane. And students, this is a very important term here, membrane biogenesis. Fine. So you can, if it comes in your exam, you can write down about the membrane biogenesis. Like there are some certain enzymes that is present on smooth endoplasmic reticulum. For example, we have lipid, steroid and cholesterol. And all these things, they basically helps in the formation of plasma membrane. And this process is said to be membrane biogenesis. Now students, another thing we have is the rough endoplasmic reticulum. It is basically concerned with the transport of protein, or we can say the secretory protein, example mucus, digestive enzymes and hormones which are synthesized by the ribosomes on their surface. So We all have seen here, students, that ribosomes are present on rough endoplasmic reticulum, and the main function of these ribosomes is to form the or to synthesize the protein which is required by the body. Another one we have is the SER, that is the smooth endoplasmic reticulum, and the function of this smooth endoplasmic reticulum is to help in the process of detoxification. Now students, through air, water, lot of impurities they comes in our body, fine. So they has to be detoxified, and here the SER performs that function. And the last one we have is, students, that digestive enzymes of lysosomes, they are also produced by the rough endoplasmic reticulum. So this was all about these two organelles that we have studied, students. The Golgi apparatus, fine, and we have the endoplasmic reticulum. I hope the topic is clear to each one of you. Thank you.